Hello, everyone. Welcome to Medicine and Miracles. I'm your host, Dr. Scott McMahon, and we're going to do something special today on Medicine and Miracles. This is the first time we've ever broken our normal format where we talk for a little while about science and medicine, and then we talk for a little while about miracles. And today we're going to interview Dr. Ritchie Shoemaker. Dr. Shoemaker is the person who discovered back in 1996 what today we call chronic inflammatory response syndrome. There is no one on the planet who has more knowledge about this illness than he does. And I have to tell you, Dr. Shoemaker, I usually call you Richie. I am so excited to have you as our first guest on our show. So welcome, Dr. Shoemaker.
Well, I am delighted to be seeing you smiling and happy. This is a great beginning for a new career for you.
A new career. That's exactly right. Woohoo. We'll see how it goes. So, Richie, do you want to tell us a little bit about yourself, your background, how you got involved in the world of chronic inflammatory response syndrome?
Well, sure. I was the third of three children. My mother was a first grade teacher, and my father was a chemical engineer, worked for Johnson and Johnson before he started working on reverse osmosis. And a lot of what we take for granted with filters and water clarity and safety and all that is due to his work. So shout out to my dad and my mom today. The family situation is that mother was born in Charlotte, North Carolina, and she would go home to have her babies. So Wellesley was the first, and Ellen was second, and I was third. And we all came out on Presbyterian Hospital in Charlotte and went to Inca near Asheville, then to Chicopee Mills and Wilbraham, Massachusetts, then Princeton, New Jersey. That was my formative year in Princeton. So my baseball team of the Yankees and the football team of the Giants, and they're not very good these days, but it's still fun to say, yeah, I'm a Giant fan. And then when I went to college, of course, that was Duke University, and you're branded with Duke basketball, as you well know. So those were the formative years. But we moved to Carlisle, Pennsylvania for my 3 years of high school. And that was when I was free with my competing siblings at home, and I could do what I want and study what I wanted. And my world revolved around playing tennis and wrestling. Um, I had taken a scholarship to Duke, and it was going to pay a full ride for the year. Back then, that was $3,500. That was 1967. That was 66. It's a long time ago, but wasn't sure I wanted to take the scholarship. So that summer, I worked on the highway and loaded trucks at the AMP on the weekends. Uh, Refinished furniture and got me used to working with wood. And it's a third job. And then we played poker with the guys. And I was tired and thought maybe because I was doing a lot. Got out the Encyclopedia Britannica, my first medical book, and says, mono, looks like that's what I have. So I went to the doctor before I went to college. He goes, yes, you have mono. You can't do anything for 12 weeks. Well, that 12 weeks was over, and I would not have been in college if I listened to the doctors. And lesson B, don't listen to the doctors when they tell you the wrong things. But anyway,
Exactly.
at Duke, I started with research and was looking at the effect of light on flagella motion of a little algae. And I thought that was great. And then my sophomore year, I started electron microscope work, looking at DNA initiation sites in tetrahymen on another protozoan we could play with. But then I went home for the one summer between my freshman and sophomore year. My dad had bought a new farm. It's an old farm, degenerate farm, 126 acres waiting to be fixed. So I said, I'll trade a year or a summer, work on the highway to help you out. I thought it would just be a little bit of work. I think I made $3 an hour, but that was good. But it got me used to use my hands and woodworking and digging in the soil, and things that kept me for well. So Uh, my father also chipped in and he said, well, how's the research career going? I said, well, it's great. It's having a great time. Electron microscope work is exciting. He goes, well, maybe you'd like to spend a day with a couple of my friends. They're over at Carlisle Hospital. Bill Shelley's a surgeon. You can walk around with him, seeing patients, and another friend was a pathologist. And a microscope. That sounds fun. But after one day, I was hooked. So much for the research career. I wanted to be a primary care doctor, and yeah, that was well. He in his own quiet way showed me what I needed to know, and so then and to Duke Medical School, and my junior year, I had decided I wanted primary care experiences, and I started writing a little journal, and it turned into a national publication. It was just typed pages sent in the mail, so it sounds more impressive than what it was. But it's a national primary care medical student journal, and we had about 2,000 pages a week, and it was called First Contact. And of course, the jokesters, and you would have been first, said, "Oh, this is a book about venereal disease, right?" And I said, "Yeah, it's First Contact. That's funny." But I went to Williamsport Hospital because they had a good residential program and rural medicine, and Williamsport was great for Little League baseball, and was lots of places to moonlight, and it was beautiful. So that's where I went for residency for 3 years, and I had already refinished a little bungalow in North Carolina and in medical school, and then went to Williamsport and refinished a Victorian home, and that was fun. Got into National Service Corps because they would cover the cost of medical experiences, medical costs. And, all I had to do was work a year for every year for them in a medically unserved area, which brought me to Pocomoke. And there I found a house that was built in 1806 and 1814. And I got to play with that. It was 5,000 square feet to refinish. And I liked it in Pocomoke, and I've been in Pocomoke since 1980. Well, that was pretty much, I got married in 1981, and my wife and I had a great time. She was a school teacher as well, and we finished the old train station. It was called the Learning Station for our program, and things couldn't be better.
And then CIRS.
We moved to Pocomoke in 1991, and it was a chance to have some land, 15 acres, and backs up to the Pocomoke River. It's a tributary of the Chesapeake Bay. And everything was great. You didn't have to lock your doors at night, as wetland experiences and great things. And then quietly in 1996, we started seeing fish with some lesions in the Pocomoke River, and these red spots, or lesions as we call them, was associated with funny motions and movements. They didn't scare away when you would wash your hand in the water. They come up and kind of float around, and the watermen who harvested these fish and crabs and oysters, you got to say it right, oysters. It's not oysters, it's oysters. But the watermen started saying they were being sick when they exposed to the fish with lesions. And there was rumors that these fish with lesions from North Carolina had been causing trouble in 1993 and 1994. But no one was sick, couldn't possibly be sick. So my friends, the watermen, were getting sick. And either I'm wrong saying they are sick when they're not, or they're wrong when they don't know what sickness is, or the politicians are wrong by saying that the illness didn't exist. So I started doing autopsies in fish and doing this and that, and came up with environmental theories. And the state had changed their mind. There was no illness. Now they said, well, the illness is coming from chicken manure on the land. Well, that didn't make sense, because what we were looking at were tomato plantations where different kinds of fungicides were used, because the new blue mold was wiping out tobacco in North Carolina and tobacco in Maryland, and tomato plantations. And they used copper and dithiocarbamates to kill the fungal elements. But it turns out that what we did with copper is kill the cryptomonads and the green algae, the Pfiesteria, the cell from hell, the thing that was making the fish had lesions. It killed their food that lived on the bottom of the ocean, of the Pocomoke River, and caused these cells to change life forms. They went from a slow-moving amoeba phase to a fast-moving feeding and breeding phase, And this is where they made toxins. So after a rain event and water was stirred up, the chemicals in the pore water or the little bit of water, if you ever made a mud pie and squeezed it, you got water that came out of the mud. Well, that's water between the particles in the mud. That's pore water. And that's where the copper was. That's where the dithiocarbamates were. And we had a 1-2 punch. We killed the organisms that the motile phase didn't care about, but the amoeba phase did. But when we killed the predators of the motile phase, we had blooms and fish kills galore. And that's when people started getting sick, and that's when I got sick. But I said, I'm going to figure this out. So sure enough, on the big day, the state of Maryland was going to have their presentation showing why there's no sick human beings. I had taken color glossies the day before. The day before, oh, and got them developed, and then on the way up to the Salisbury for the conference. And the Lewisman, when they saw, here's my pictures, this is what the lesions look like of the sick people. Made me real popular, I'm sure, but it was off to the races because then what to do for the sick people. And about the day 3 or day 4, the news was all over this. People like getting my name. And one young lady who I'd known for a while came in. She says, well, I've got that lesion thing. I said, oh, do you have a lesion? No. Well, what do you have? Well, I'm tired. I hurt all over. I can't remember things. And I got some damn diarrhea. I've been going 6 times since Tuesday. And I said, oh, wait a minute. Diarrhea. Did you eat anything before the diarrhea? No. So it's secretory diarrhea. Oh, well, I'm an old family practice veteran. Here's cholestyramine. We use cholestyramine to bind bile to stop secretory diarrhea. And 2 days later, she calls up. She goes, well, thank you. I feel great. Then the medicine worked perfectly. My diarrhea stopped. And I said, well, how's your headache? Oh, that's gone. How about your cough? Oh, that's gone. How about your aching? Well, that's gone too. Now, when I think about it, it's all gone. Just how can that medicine have done that? And so I was off to the races with 200 patients and wrote the first paper on diagnosis of Hysteria human illness. I called it FISH. I thought it was cute to have an acronym that would make a word, but CDC changes it to PEAS, possible estuarine-associated syndrome. And like with chronic fatigue syndrome, they kind of emasculated the disease by giving it a girl's name. But anyway, PEAS and FISH, it was fine. But then I published a paper in September of 97 on diagnosis and treatment in February of 1998. And the phone started ringing from Florida, wanted to know they had a dinoflagellate outbreak like this was. They had a blue-green algae outbreak like this was. And one thing led to another. Woods Hole Institution wanted me to treat ciguatera, which is another dinoflagellate illness. And, by that time, the EPA got involved, and Ken Hudnell had a vision test called visual contrast sensitivity. And he was able to show that where people sick were in North Carolina, they took his test, they would get sickened. They took my treatment, the sickening would go away, and the vision test would get better. But then they got sick again, the vision test got bad again, and then we got treated again, the vision test got better again. We were doing repetitive re-exposure trials back then. So, but anyway, in 1998, there was a phone call with a woman in Princess Anne who had all this black stuff growing in her mold while the water leaks were. And she was sick as the dog. So I went up and sampled the black stuff. It was Stachybotrys. Didn't know much about it, but it was the organism that scared people about fungi. But the VCS test was positive, and cholestyramine helped her feel better. And that was great until Lyme disease came along. I treated a lot of Lyme. People would come all over the state of Maryland to get treated in Pocono because I didn't think the disease was folly. I thought it was real. But when I gave cholestyramine to Lyme patients, they've been treated with antibiotics. Oh my God, did they get sick fast and severely? Oh, it was terrible. It was like the sudden violent wind blowing the blossoms off the pear trees in springtime. That's my image of this cytokine storm that we're getting with Lyme disease. But that was the first indication that cytokines were involved, and I still didn't know why some people got sick and other people didn't. And found out about HLA-DR by PCR. LabCorp would do the test for me, And this genetic test on chromosome 6 controlled immune responses. And if you had a certain gene makeup, you'd get sick. If you didn't have the makeup, you didn't get sick even if you're exposed. Well, now we got cytokines. And how about regulation of cytokines? Well, there's MSH, melanocyte stimulating hormone. And those are my first key findings. And then it didn't take too much longer. We started, VEGF came along, and TGF beta 1 came along, and MMP9 came along. And I had a new career. Went from a family doctor and, seeing 45 people a day. And if I made people mad at Walmart, they'd tell me about it and they wouldn't come see me. But, I've only had 40 go, I made somebody mad, and that was not unusual. But it was fascinating to see how this whole idea of organisms that made toxins could be treated by a compound that wasn't absorbed. So the compound is in the duodenum. The toxin was in the duodenum, but people stayed sick. So it was reabsorbed from the jejunum and the ileum from the duodenum. So we have enterohepatic recirculation of a biotoxin we can interrupt. And I wanted to get a stool sample. I want a stool sample to look at Listeria in the stool, in the toxin, with cholestyramine. And I had 7 freezers full of stool before Barb Gray finally, who was working for me then, says, don't you think we ought to clean out some of those freezers? You haven't done anything with it in 10 years. And I said, well, I always wanted to have fun. I always wanted to have freezers full of stool. And I think I used a different word for it, but it didn't matter.
Poop.
It was about 2010 that you and I got together. You had worked in Roswell, and we had a book that we were postulated. It was not even a hardback book. It was just paper bound, but it's all designed about policyholders of America and CIRS, and we named the illness CIRS. It wasn't my credit; it was our credit, and no one gives you credit except me. I don't know why that is.
I I don't need any credit, Dr. Shoemaker. You have done so much to define this. And at this point, we probably need to take a break, and this will be the end of part 1, and we'll come back and discuss some more about this. If you want to see Dr. Shoemaker live, if you want to see him in person, don't forget to check out the Surviving Mold CIRS X annual conference that's going to be held in Tempe, Arizona this year. It's June 5th through June 8th. You can come virtually or you can come in person. The hotel rates are amazing. They're the best we've had in years. It's an amazing facility. Check it out at cirsx.com, www.cirsx.com. Today, we have part 2 of the interview that we're having with the person who discovered chronic inflammatory response syndrome, Dr. Ritchie Shoemaker. Thank you. Welcome for being with us today, Dr. Shoemaker. How are you doing in Pocomoke?
Well, we're going to have some sleet and freezing rain and snow tonight. This is almost unheard of. This year has been crazy. January was polar vortex time and cold, and now we've got more crummy weather. Yesterday it was 60 degrees. Well, this isn't right. I should be outside digging the dirt.
Yes, you should. Yeah, we've had some crazy weather here in Roswell also. We're in the middle of the desert and we had a flood about 4 months ago. Who knew? Oh wow, it's probably all related to dinoflagellates and the balance of toxins, but I'm just kidding about that. Hey, you were telling us about how you discovered CIRS and kind of the progression of things that happened. How many biomarkers would you say that there are for chronic inflammatory response syndrome now?
We started with none, just simple clinical observation. We added visual contrast in 1997, so we had 1. We now have 50 published biomarkers. 50.
That's just jaw dropping. I think 50. And, I've read the reports and by God's grace, I've gotten to author some of them with you. The p values on those, the likelihood that these are incorrect are pretty astonishing. I mean, the science that you've put together over the last nearly 30 years is really astonishing, and I tip my hat to you, Dr. Shoemaker.
Well, the nice thing is, in large part, I've been able to work with people like you, and a tip of the hat to you for your diligence and your skills and your humanity. You've been the best friend I think a friend could ever have. So, uh, more than a hat to be thank you. I'm getting gray hair, and yours is not, so you're going to have to take all this work.
you you warm my heart. Thank you very much. Um, it's a lot of our audience are patients or lay people. They don't have medical background. If you were going to explain what chronic inflammatory response is, how would you go about doing that?
Usually, this conversation comes up when someone says, "Well, my aunt has tired all the time. She's just not thinking right. They think maybe she's got early stages of dementia." Someone else will say, "Well, uh, I've I've had Ciguatero on a cruise. We went on a nice boat, and they said it was safe to eat the fish, and it was not. It was grouper and barracuda and snapper, and we all got sick." And somebody always knows someone with a biotoxin illness because the people are tired. That's the most common one, is this multi-system, multi-symptom illness. They've got cognitive issues and respiratory problems and pain and headaches and neurologic problems and gastrointestinal problems and skin problems. And then you say, wait a minute, this illness is systemic and it's chronic, and it doesn't get better with antibiotics. It doesn't get better with psychiatric medications. It doesn't get better with rest. In fact, it starts to get worse. You get more and more problems with cognitive issues as time goes on. And if you moved out of the house that you think made you sick, how come you weren't better? I mean, if you had an allergy, you'd get better. Well, CIRS is not an allergy. If allergy is a part of the immune system, call it the right-hand side, if you will. The left-hand side is innate immune responses. Allergy is based on manufacture of antibodies, and then these innate immune receptors are sending the word out to the body that a inflammatory stressor has arrived on the front door, or through the nose, or through the GI tract, or through the skin. But those differences, if you look at IgE, you find that a lot in allergy. IgE is normal in chronic inflammatory response syndrome. And inflammation is created by these innate immune responses. But we now know that metabolic disturbances are present, and abnormalities in manufacture of DNA and RNA and proteins by ribosomes are crucial in this illness. And ribosomes were the things you learned about in biology in 10th grade in high school and promptly forgot about it. Because who wants to worry about ribosomes? But we don't forget about ribosomes because they make proteins. And then mitochondria, the powerhouses of the store of the cell, their DNA left mitochondria a long time ago, went into our nuclear DNA, where they're controlled by nucleotranscription factors. And if we disrupt normal manufacture of RNA from the mitochondrial DNA, we're going to find out that metabolism is not the same as what we want. And here we've got sugar or glucose getting into cells the way they're controlled by glucose transport proteins 1 and 4. With the delivery boys bring sugar into glycolysis. Glycolysis breaks sugar down and makes 3 carbon fragments instead of a 6 carbon fragment to start with. They're called pyruvate. And pyruvate is carrying the energy codes into the mitochondria. But if there's the DNA problem, the transporters that move pyruvate out of the cell into the mitochondria don't work right. The outer cell becomes a wall, not a pore. And so what's the cell going to do with all this pyruvate? It can't be used for energy. We don't make ATP anymore. Maybe a little bit of carbon dioxide and water is all we get from pyruvate, but we can make it into lactic acid and convert lactic acid into something we can excrete. but we create an illness called lactic acidosis instead of normal energy and normal protein production. We've got another biomarker. As we follow what lactic acidosis will do, it will cause abnormalities of T regulatory cells or T white blood cells that regulate inflammation. It'll change pulmonary cells and into ones that create pulmonary hypertension. And who wants to walk around feeling short of breath all the time? It's not your heart, it's your lungs. It'll change brain areas called gray matter. And these nuclei, these centers of gray matter, get destroyed because these compounds, these toxins, can travel through tubulins. And who's heard of tubulins? These are the predominant carriers of ions and nutrients in parts of neuron cells called axons. And the axon should feed the synapse, but it doesn't feed the synapse if the tubulins are not working, if there's metabolic acidosis. And then we come along, we fix the tubulins, we fix the acidosis, and we fix the synapse, and we fix the nuclei. And suddenly the brain that had 3 areas or 4 areas or 5 areas or 6 areas of problems, and never was 6 areas were corrected at once. Suddenly our patients, in a paper you and I wrote in 2017, we were able to show our brain atrophy was resolved and equal to that of controls. How about that for something to take from pyruvate?
That's an amazing uh, an amazing sequence of steps that you started with, just from a cellular level throughout the body, but then ending up causing abnormalities in the brain and cognitive issues and whatnot. That can be seen on special types of MRIs being done of the brain, and all of that as the father of that science. So pretty darn amazing. Thanks for that.
Well, you're welcome. You know, what's interesting is that after you and I have treated these patients and they've got their brains back, of all the CIRS patients in the world, the hundreds of thousands, the millions in the US, they're all just diagnosed improperly. But of all these people, the thing they miss most is the brain.
If you saw I'm sorry, if somebody is watching this and they were struggling with maybe some brain fog, fatigue, maybe their muscles or joints are hurting, they're tired all the time, they're getting headaches, how would they find somebody who could help them? How could they find a doctor or a nurse practitioner or some provider who actually knows about chronic inflammatory response syndrome and could benefit them?
Probably the easiest way is go on the internet and type in CIRS.
Okay. What about surviving mold and their interactive chart?
You'll probably have a chance to see surviving mold pretty quickly listed on the internet. There's a listing of physicians who have been certified and physicians who are diplomates or proficiency partners who are ready to see you. It's a lot easier now than it ever used to be. Scott, you've got a website called CIRSX that has a list of physicians, doesn't it?
It's developing a list of physicians, and it's also developing a list of indoor environmental professionals that can do mold testing or remediation who are familiar with CIRS.
We'll hopefully talk about that. It's a big problem is that remediation has got some people that are doing things in state of the art, but most people who are doing remediation are still doing air samples and things that went out 25, 30 years ago. So there's a new world of sick people from water damaged buildings that we talk about quickly. We're just saying, oh, sure, it's CIRS. But the IEPs are the ones that are making the difference in making the buildings safe.
Thank you, Dr. Shoemaker. This is episode 2, part 2 of our interview, and we'll be back with more. If you want to see Dr. Shoemaker live, if you want to see him in person, don't forget to check out the Surviving Mold CIRS X annual conference that's going to be held in Tempe, Arizona this year. It's June 5th through June 8th. You can come virtually or you can come in person. The hotel rates are amazing. They're the best we've had in years. It's an amazing facility. Check it out at cirsx.com, www.cirsx.com. And today we have part three of our interview with the person who discovered chronic inflammatory response syndrome, Dr. Ritchie Shoemaker. Dr. Shoemaker requires no more introduction that if you know about CIRS, you know about Dr. Shoemaker. He's like the godfather of CIRS. Okay, maybe that's not a nice thing to say, but he is the person who's discovered, most of the pathophysiology and most of the treatments and most of the biomarkers, if not all of them. So, Dr. Shoemaker, thank you for being a part of this podcast.
Well, I'm delighted to hear you speaking so well, and especially you letting me be on your first guy on your show. My goodness, that is a real honor. Thank you.
Our very first interview is with you. That's right. We actually saved up. I mean, we waited there. There were other people that actually asked if they could be on the podcast. And we said, no, we're not going to interview anyone until we go. I was going to say to the horse, but that's not quite right. To the horse's
That's more
mouth.
apprehension's head, right?
Exactly. Exactly. So when we left off last time, we were starting to talk about the indoor environment. And of course, the indoor environment, the built environment is where the problem is with most people who have chronic inflammatory response syndrome. They get water damage in their home or in their office or at the school that they might be attending. And you were starting to talk about, what some of the problems are in that industry of detecting mold and fixing it. So take it away from there if you'd like.
One of the things that's important to recognize is that all wetness inside buildings is not the same. If we've got a flood of cold water, a flood of warm water, if you've got a hurricane, you've got trauma with that, you're going to have different injuries to the ecology, the living environment, is what's inside a building. If the building has been dried out and you're then re-wetted, if it's never dried out at all, you're going to see organisms that by their presence identify the source of the problem. Now, the physics term of activity of water, or AW, not A&W root beer, but activity of water, looks at a beaker of water, distilled water, and the amount of water that evaporates out at room temperature is going to be a percentage of the water that stays. So if we assign the amount of water that evaporates or dries out of the beaker as 100%, we're going to find that at 90% is where we find the wet-loving fungi. So Stachybotrys, I had mentioned back in the first talk of this series. Chaetomium is a wet one. Bacteria are wet ones. And if you find 90 to 100%, that's going to be usually a fresh flood or fresh water event, fresh plumbing break in the kitchen, fresh shower over leak, but fresh leaks of water anywhere in the body, excuse me, in the building. But specifically, it's not going to be the same as the basement and the crawl space, because there the water coming out of the air will be a little drier over time, and its AW will be a little lower. So that we'll find, in fact, some of the driest fungi that we have, Wallemia seebi at 0.55 for activity of water, kind of defines why we want the indoor environment and the indoor humidity to be less than 55%. We don't want to give water for the ecological niche of a given species of organism. Now, we also have penicillium, we have aspergillus, two aspergillus, one aspergillus penicilloides is a real bugaboo. It likes AW to go between 0.65 and 0.85, so it's broad ranged. Fortunately, penicilloides, is pretty easy to eradicate just by blowing a fan on it. But Aspergillus versicolor, another Aspergillus species, and you can't tell the difference between the two by looking at them in a microscope. Same with penicillium, you can't tell the difference between that and Aspergillus in the microscope. But Aspergillus versicolor doesn't like 0.65, it likes 0.8 to 0.9. So you can start to stratify the ecosystem having organisms based on the water content. But we also recognize now that organisms that let fungi grow are creating a niche for organisms to break fungi apart. Actinobacteria make glucanases, and beta glucans are part of the cell wall of fungi. And if you don't find fungi, but you find a lot of beta glucans, be looking for actinobacteria having dissolved the fungi. Actinobacteria like drywall and cellulose, that is alkaline. Fungi like drywall, that's acidic. These guys are in warfare. They're balanced warfare in the body. We use the acronym FAB for fungi, actinobacteria, and bacteria that are found in every oral cavity and cavity that there is in the body. So in the gut, in the GU tract, in the GYN tract that we were worried about, in the mouth, in the nose, you're going to find actinos and fungi and gram-negative rod bacteria. Well, the question is, if you've got these organisms in you, why doesn't everybody have the illness sick? Why don't they all get CIRS? Well, the answer is that the HLA is telling us who's going to be making an innate immune response with no antibody response. And this antibody response is what separates innate immune from allergy. And if you don't get an innate immune response, you're not going to wipe out the offending allergen, which will start taking into chances with fungi receptors. And then the receptors were going to be bound to antigen presenting cells. They process and put HLA on the receptor marker, and then they present to a naive T cell that says, okay, here, B cell, make your antibody, away you go. In CIRS, we don't get antibody production. So we're working backwards. Does that mean that the people that get sick from these FABs, from freaking out, are because the FABs are out of balance? Yes. Or could it be because they've got a genetic susceptibility? Well, that too. So the basic idea is that if we can identify what's wrong with a building, we can identify what's wrong with the overgrowth. We can identify with remediation what you do to clear out the building and make it safe. What we do with fungi is to remove areas of damaged drywall and damaged cellulosic period kind of structures. We also will remove 2x4s and joists and wood if we have to, if it's coated with microbial growth and you can't paint over it to clear it up. So that's one thing, is that fungi has remediation, but you have to do something very different for actinobacteria because the substrate is alkaline, not acid. And the same thing we also true with bacteria, because they will be ones that'll be making a smell like actinobacteria. And that's what we're warning about a water damaged building. And, the experts will all say, if you get musty smells, be worried about fungi. I say, wait a minute. Fungi aren't making the musty smells. Actinos are making the musty smells.
And it's geosmin.
Bacteria are making the musty smells. Why don't we worry about them, and why are we worried about fungi? But the point is that what you do to clean a house is dependent on what's wrong with this house. I'm not an environmental professional, and Scott, you might have more training than I do, but we have, fortunately, made connections with outstanding indoor environmental professionals who can be brought in to clean out houses, not for burning them down, not for spending $100,000, but by doing things right. So you're not going to be saying, well, what did the results show of all the cleaning you did? Oh, you didn't do any follow-up studies? How would you know if you got it all if you don't test for it? And then specifically, how would you know if what you did didn't work? And this was the little key to the literature that came out of Finland, which we used as our first actino-based paper, because there they studied buildings that were remediated the best of all possible ways, and it still made people sick. They got rid of the fungi, but what else was there? Actinos. And we now can identify actinos. Hopefully we'll have a chance to talk about Jeannie and section number 4. If we want to know specifically what's wrong, what made the building make people sick, there's a specific makeup that will tell us about the actinobacteria, and the specific causation is one of the term I'm after. And you'll have specific causation for endotoxins, if endotoxins made by bacteria are the source of the problem. If fungi are the problems, they have their own specific causation. We can tell with transcriptomics alone. Now, this test is not readily available. You can't get it done at LabCorp. But if you want to know what really is wrong, the test called Genie will give you a specific causation, and that's what you do want. Genie will give you a lot more, and we could talk about that another time.
Yeah, we should. Do you think that most of the remediators throughout the country, I mean, these are the people that are going to come into your house that has water damage and mold, and they're going to try and remove all of that material. Do you think most of them are aware of what you're talking about? That that there can be problems with mold as well as actinobacteria as well as bacteria?
Based on what I see on a day-to-day basis, the answer is yes. Now, my answer is biased because I get the complaints. I get the things that people don't have. I hear the people upset about having spent money they wasted because the problem was not fixed. But specifically, if we look at who is not getting complaints, these are the IAPs that will identify the organisms, they'll identify people with visual contrast abnormalities, they'll identify the illness symptoms as shown in their paper published in 2021, which is something required reading for all IEPs and all patients alike. You can get that on Surviving Mold as a free download.
Right. And my experience is because I only recommend a certain few indoor environmental professionals throughout the country that I know are familiar with this, I don't get a lot of complaints either. But from patients that have had remediation before that I've come to know them who didn't get referred to that, I found that a large number of them end up maybe getting, maybe having to do a remediation a second time, or maybe there were water intrusions that were missed or things like that. Do you have that same kind of experience or did you when you
Well,
were active practice?
we have one of the concerns is what about other exposures besides the home? How about airplanes?
Yeah.
Well, the air is supposed to be turned over every 45 minutes. And as of 10 years ago, that was not the case. They may have changed to every 45 minutes now. But then hotels,
Yeah.
the hotel looks beautiful, all clean and scrubbed and all this other stuff. Well, you don't know what happened a week ago to the kids playing in the bathroom while mommy and daddy at the conference. They're making 2 floors of rooms wet. They get cleaned up and you never know it. So,
Cruise ships.
excuse me, and cruise ships.
Cruise
Yeah.
ships.
So I mean, and one of the other things is, Scott, I know that you talked about is what really are the most important things that a patient should do. And that is if you are worried about your living environment, do the testing. Envirobiomics in San Antonio will do the testing. Micrometrics in New Jersey will do the testing for you. But ask them, do you test for endotoxins? Because endotoxins cause, we were talking about uh, neuroquants and abnormalities in the brain. The worst patterns of illness and worse injury to the brain are caused by endotoxins. And it looks like the endotoxins can cause their damage 6 months later. You find injury that's not healed by removal from exposure. And so if you find someone sick for 12 years, you think of endotoxins as their initial source of problem. Endotoxins turn on inflammation in the brain and microglial cells and all that good stuff, and toll receptor 2, and boy, state of the art. But the whole issue is that if we don't know what's wrong, we won't know what to take care of it. And if you do the testing right and rehearse for what you're going to say to the IEP guy that you want to work on your house, I need to make sure that actinobacteria are cleared out, and don't use an air sample for me, because get out the World Health Organization publication of 2009, which says specifically stop doing air testing. Doing 5 or 10 minutes will tell you what's in the air, but not tell you what's settled out. Won't tell you what else is the aspergillus or penicillium. You can't tell the difference. Won't give you Wallemia, won't give you stachybotrys. There's not much point in paying for air sampling at all.
Yeah. There's a study that came out of Houston in the, I believe it was in the late 1990s, that showed that 85% of homes that were tested for Stachybotrys, when they found Stachybotrys by other means other than spore testing in the same room at the same time, that the spore trap testing missed that Stachybotrys. And of course, that's the mold that the lay people are all concerned about. Toxic black mold. Oh no! And yet spore trap testing misses that at least 85% of the time that it's there. Unbelievable. That's all the time we have for this segment. Thank you again for your time, and we're going to conclude episode 3 or part 3 of this interview, but we've got more to come. Don't forget to check out the Surviving Mold CIRS X annual conference that's going to be held in Tempe, Arizona this year. It's June 5th through June 8th. You can come virtually or you can come in person. The hotel rates are amazing. They're the best we've had in years. It's an amazing facility. Check it out at cirsx.com, www.cirsx.com. This is part 4 of just a really enlightening interview that we're having with Dr. Ritchie Shoemaker. He's the one who discovered chronic inflammatory response syndrome, and he is a repository of knowledge. Dr. Shoemaker, is at the end of our last segment, we were talking about actinobacteria and endotoxins and whatnot. And did you know that at the upcoming Surviving Mold CIRS-X annual conference, the one that's going to be held in Tempe, Arizona, June 5th to 8th, did you know that one of our speakers is going to be talking specifically about sewer gas?
That would be Eric Dorninger, wouldn't it?
He is gonna talk about that, but we have somebody who actually specializes from the Arizona region, who specializes in sewer gases and the endotoxins that come with them and whatnot. I believe his name is Derek Denis. It's spelled like Dennis, but it's pronounced Denis. I haven't met him, but I'm excited to hear what he has to say.
Well, I would much rather hear him when he's talking about sewer gas than Eric passing gas, quite frankly.
Well, and I heard that he has a real potty mouth, so we'll see. And you're going to be speaking also. What do you recall what your topic is?
Well, I'm going to the brain. I've got Parkinson's disease, and I'm doing not so much better with it. I want to prove the science. Had my visit with a neurologist yesterday, and gave him a paper we've just written, and we'll have another new one by Tempe time, looking at the mechanism of brain injury leading to Parkinson's. It looks like we've got a winner, and if we can clear endotoxins out of the equation, we'll have a lot of people not getting Parkinson's disease.
Oh, that's fantastic. And of course, Dr. Dale Bredesen will be there talking about, his work with Alzheimer's. But let's stop with Parkinson's for a second. And I know that the work that you've been doing that is through Genie, and you mentioned Genie before. It's a transcriptomic test which looks at what's going on in the cell. Why don't you tell us a little bit about what you have already discovered, through Genie about Parkinson's and exposure to mold and the like?
We have 200 genes that we manage to test. We need frozen samples sent to the lab from in Boston. Your physician can draw the blood, make sure it sits up for 4 hours, and then refrigerated and then frozen. Once it's frozen, it can be mailed. But what we found is that 85% of people with chronic fatigue have got hypometabolism. This is the injury to the ribosomal production mechanism of proteins. We now know the basic physiology of all this. If you want to know one thing wrong is there hypometabolism? Secondly, is there disruption of normal energy production mechanisms? Thirdly, do we have proliferative physiology creating lactic acidosis and leading to all the problems with pyruvate that are so commonly found in CIRS? We've been able to show defective apoptosis and regular apoptosis. We've been able to show apoptosis caused by fungi. We've been able to show apoptosis caused by MAP kinases. It's all fancy names, but they're all different. It's like the biology of the ecology and the pathophysiology of the illness. We have looked to see one new finding, and that is we think is a fingerprint for Parkinson's disease. We'll have CLU, which is one in the packet or the venue for apoptosis. We'll find CLU. Under coagulation, we'll see where Eliquis can make a difference and other anticoagulants can help. We'll find 6 or 8 genes upregulated in coagulation, and they all have roles to play in the microbiology and the microanatomy of immune response. We've been able to show cytokines have particular kinds of effects and presences. We're able to show specific causation of actinobacteria, both from TGF beta 1 receptors, TGF beta 2, TGF beta 3. These receptors, in combination with MAP kinases, give you specific causation. If we look at endotoxins, we'll look at CD4, TOL2 receptors, TOL4 receptors. We'll look at fungal causation with the MAP kinases and the fungal caused apoptosis. And all this is one blood test. But the TOL2 receptor is the real packet of interest right now, because this is one that will turn on microglial cells to start destroying more of the gray matter in the brain and more of the white matter in the brain. And we look at Parkinson's particularly, and the microglial cells will spread alpha-synuclein, the king bee of Parkinson's, from cell to cell, and destroy autophagy, which is the cell's mechanism to destroy alpha-synuclein. But if we can control microglial cells, which we can stop the illness. We're now looking at this pattern of antigens, excuse me, of genes present in kids that are 4 years old. You were seeing children who had Parkinson's genes and didn't know it. Would you have treated that child for Parkinson's disease without Parkinson's symptoms? Well, we're looking at the DNA mechanisms and the genomic mechanisms that lead to Parkinson's. Why are we waiting 40 years for the illness to be reproducibly reliable and killing people? Why aren't we treating it while we still can? These are questions that will be answered, and I hope the TOL2 talk that I give will give some answers onto what is ahead in the world of Parkinson's. When you ask what is the role of Genie in the future of medicine, it's going to be telling us what's wrong before we know what's wrong, so we can fix it, and then it won't be wrong.
Very cool. And CLU is clustering. And did you want to mention anything about the tubulins also?
Thank you for mentioning that. The third element of the triple positives are cytoskeleton tubulins. These are the ones we talked about with microtubules that are found in axons that deliver the nutrients and ions that the cells need in the neural body. Every cell has got tubulins in them. When the cell is dividing, tubulins will attach from one pole of the cell towards the collection of those chromosomes called centromeres in the middle of the cell, and will pull the DNA to make 2 daughter cells. So cell division, you need tubulins. If you've got defective tubulins, will you have correct cell division? No. This is where the problem with fungi started getting known worldwide, because the antifungal disrupts tubulins and disrupts its attachment sites in centromeres. So we can create mutants that survive the antifungal, but are no longer have the right DNA, and we're going to be making genes that are abnormal, found in organisms that didn't live inside buildings until they were poisoned and mutated, and now they do live in buildings, and now they're everywhere. I would rather be in a Scottish castle 1,000 years old than the newest apartment complex in Salisbury on the Wycomico River.
Wow. It's not every day that I get to talk about mitosis with people. So thank you for taking me back like 45 years or
And meiosis too. Don't forget that.
and meiosis, because you got to get the germ cells too. Exactly. So I'm going to switch gears a little bit, if I might. Uh, if you were talking to a CIRS patient today and they were new to treatment, what would you say? and I got to stop here because the only peer-reviewed published method of treating chronic inflammatory response syndrome that I know of is called the Shoemaker protocol. So, thinking about the Shoemaker protocol, which you probably know something about, I don't know, it wasn't the, yeah, we'll just say that maybe you created that and tested it and published.
Yes.
Yeah. Uh, Based on that, what would you say are the most important steps? I mean, they're all important, but what are the most important steps for patients to get right?
I think steps 1 and 12. There are 12. The first step is removal from exposure. And how you know you're exposed? Well, you can test your house. You can look. You can smell. You can have a water event and you know it. But then how about the hotel that you don't know about? You have to remember that simple exposures, even for 10 minutes, just smelling geosmin, the actinobacteria smell, well, it can be a warning that it's time to get out if you're genetically susceptible. So if you're a CIRS patient, step 1 is removal from exposure. Having said that, step 12, if you go through 2 through 11, is use of VIP to do the cleanup, to do the finish work, to do the completion of the reparation of the brain and the reparation of the immune responses. So those two are the two biggies. I hesitate to leave out number 3, which is correction of Marcons, bacteria that are commensals living in the nose, because if they're there and they're thriving, you will not get better just by using step 1 and step 2. So step 3 is important. You can find step 1 through 12 in the physician essays on surviving mold under the What is the Shoemaker protocol. The 12 steps are written right out for you. In fact, Scott, your essay was one of the best still. You were the first and one of the best.
Well, thank you. Well, I vividly remember talking to you in your office back there in 2010, and you telling me that for the vast majority of people that don't get better, there's only three reasons. Three. The first one is they're not taking a sufficient amount of binder. The second is that they have untreated Marcons. And the most important and the most common is that they haven't gotten out of their exposure yet. And that's steps 1, 2, and 3 of the Shoemaker protocol.
15 years has gone by since we had that conversation. Don't you think we should have learned something more by now?
Ah, well, I still haven't seen anything, that has been frequent enough to be a fourth reason. Those three pretty much always cover it.
Well,
That's
you're right.
2,000 patients for me later.
We have created enough biomarkers now. We've gone beyond 3 steps. But if you don't do the first 3 correctly or don't do them down the road, you won't get better. The problem is not the protocol. The problem is 1, 2, 3.
Yep. When we come back, I want to talk about the use of systemic antifungals and see if I can get you riled up at all. So, thank you, Dr.
Yes.
Shaker. We are just so blessed to have the opportunity to continue the interview that we've been having with Dr. Ritchie Shoemaker. Dr. Shoemaker is the one who discovered chronic inflammatory response syndrome back in 1996, and he has created the vast majority of the science that's been involved, that and how we make the diagnosis, how we treat, and how we think about this illness. He's developed 50 biomarkers over the last nearly 30 years, and he's here to talk to us today. And Dr. Shoemaker, as we finished our last segment of this interview, I just kind of brought up the provocative subject of the use of systemic antifungals in the treatment of patients that have chronic inflammatory response syndrome. Tell me your thoughts on that.
Well, there are other people who treat the syndrome, and some are treating differently than I do. The problem of antifungals has come up to be it's a real bad idea for several reasons. Number one, the illness is not a fungal infection. They're not fungi sitting there making toxins in the sinuses, creating CIRS. That's just not the way that works. Secondly, the paper I published in 2019, looking worldwide at use of antifungals and in presence of mycotoxins in urine, I published 40,000 patients from 20 papers. where the controls or well people had mycotoxins in their urine. Uh, we know full well that if we look at the work of Michael Solyuk from Austria, that if you've got a water damage building and you assay for mycotoxins, he'll find over 500 species, 500 different mycotoxins in a room. Here you're looking at 5 or 6 mycotoxins, thinking that that does something, and they're not. The issue is that mycotoxins can create metabolic problems. They destroy the mechanism to carry pyruvate across the outer mitochondrial membrane. We talked about that previously. And there's more. The voltage-dependent anion channel is disrupted by mycotoxins. But the whole issue is that of the sick people that we find, 7% have fungal specific causation. 28% have got fungal, you've got specific causation from bacteria, gram-negative rods, and 42% fungal, specific causation is coming from actinobacteria. These organisms, they make 20,000 different compounds. They're the biggest species of any, the biggest genus of any species in microbiology, from tuberculosis to cyanobacteria to all these different compounds that we use every day. Actinos are our friends, but they're also our enemies. And killing mycotoxins makes no sense because mycotoxins are making people sick.
That's well said. I can't think of a better argument than that. The last question I have for you about CIRS specifically is, where do you see the future of this illness going?
Every day that we see a new brain image from NeuroQuant is another day closer to stopping brain injury. Every day that we have a new report for Jeannie with a new fingerprint for Parkinson's disease, every day we have specific causation one more time, we are closer to finding the full answers to make people well quickly and efficiently without leftover damage from the years and years and years of illness they might have had. We're seeing already the branching out of what we've learned from uh, the problem of Th17 Treg imbalance. It's a big problem for cardiovascular wise. We know that this is created uh, by CIRS. We know that we can treat this and take care of TGF beta 1. We take care of Th17 Treg imbalance. We can make sure that we've got correction of the dieback degenerative CNS disease with the cytoskeleton TUBBA1 and TUBA4A. All of these advances in brain science are happening faster than we can keep up. We're going to be marketing more to the public to let them have access to physicians and trained individuals, nurse practitioners especially, who can help them on the telephone. The expansion of public awareness and expansion of public knowledge based on ready access to providers is going to be the biggest step forward. I think that you're involved in some of that project getting started, and I hope it's happening by the end of February, maybe sooner. But the advancement of knowledge is the advancement of science, and rigorous science and not anecdotal studies is the way we've got to go. We're following that to the hilt so far. That's why we've got 50 biomarkers, many of them from Jeannie, many of them from cytokines, many of them from laboratory findings elsewhere. But the new world is here today. We're not guessing anymore. We've got data that's been published and peer reviewed for 30 years, and Scott is leading the charge with CIRS-X, and this is so wonderful to have this new program, this new podcast ready to go.
Well, it certainly is a change of pace, but hopefully I'll have the opportunity to impact more people this way and to spread awareness. And it's really amazing, Dr. Shoemaker, where CIRS has come from, and you kind of summarized that for us earlier, and how big it has gotten, how much awareness that there is and is coming. You mentioned, www.moldco, which is, providing direct to consumer services right now in just a couple of states, but hopefully by the end of the year we'll be expanding to almost every state. The research possibilities are amazing, and all of this, I mean, is on your shoulders, and there are others who are standing on top of your shoulders who will see farther, kind of like Newton described. And it's very special to me to have had the opportunity to work with you over the years as a mentor, as a father figure, as a researcher, as a colleague, as a friend. It's really been tremendous, and so thank you for all your contributions. I was hoping at this point that we might switch the the direction that we're going. And I you know that I'm a man of faith, and we call this medicine and miracles because we talk about scientific things, but we also talk about the miraculous and the supernatural. And I was wondering if you wanted to talk at all about how maybe faith has impacted your work, or miracles that you've seen, or anything like that that you'd like to bring to our audience's awareness.
For years, my then bride would go to a Methodist church and tithe routinely to the Methodist church and sing in the choir. And sometimes I went if she was singing a solo or playing a solo. But God wasn't very important in my life. When she contracted breast cancer and died, I was left alone, literally. My daughter was nearby, but alone. And long messages of thought. I questioned my own faith. I had left the Episcopal Church at age 12. I was upset the priest would not let the youth group that I headed up sell coffee and donuts after the service so we could set up a trip to go to see St. Patrick's Cathedral in Washington, in New York City. And I said, well, the heck with that. And religion was not important to me in my state of self-awareness. So I recognized that I had cut out one of the biggest parts of my life that I used to have. And I was in the process of becoming the prodigal son myself, turning back, started experiencing spells where I would be speaking and then suddenly couldn't speak. I had spells I would, same time I'd be getting goosebumps and a sweat and have some abdominal discomfort. And when I would come out of it, it wasn't a seizure. There was an idea that I was looking for, been searching for, that suddenly I had. A woman came by looking at my house, thinking she might want to buy it. She wanted a 15-acre plot, and what was I going to sell mine? Well, let me show you. See, you might like it. Well, she was very nice, and we had a nice talk. And I said, "Can I see you again?" She goes, "Well, I'm going to be in church, so I won't be there, but I can see you Monday." And so, what about this church thing? And what about faith? And Cheryl has been my teacher and my mentor. The preacher of the church I go to now is Greg Fentress. He's written a book, and on the way home, the road to recovery. And I have discovered faith that I had and lost. I found Jesus as my personal savior. I found God as the person I talk to every day. I pray every day. I have miracles more than I can count on in CIRS. An 8-year-old girl was blind. She had uveitis associated with juvenile rheumatoid arthritis. She's going to see a specialist out west. And I said, I want a TGF beta 1. And he says, what do you want a TGF beta 1 for? Nobody gets that. I said, there's 75,000 papers on TGF beta 1. It's associated with her blindness. She's got TGF beta 1 problems, and she's blind. We'll fix them, and we'll see what happens. Well, her TGF beta 1 was 30,000. Back in the normal range was 2,300. Fix your TGF beta 1 with losartan, a blood pressure pill, and you give to children. Right, sure. And 6 weeks later, she could see.
Wow.
First miracle.
Hallelujah.
It's been one a month. I used to say we're teaching new mentees that you will have the blind see, the lame walk, and the deaf hear. Didn't realize that was from the Bible, but it is. A lot of it I didn't realize from the Bible. I never read it. I read it now. At least a lot of it. Got more to go. But I have surrendered my arrogance as being the scientist who can research. There's a higher power. There's a God that looks over me, who's the Alpha and Omega. And it's not Ritchie Shoemaker. It's humbling to be surrendered to God.
Well,
That's where,
the impacting every area of your life, doesn't it?
yeah,
Yeah. For the better. For the better.
July 23rd, 2023 was my conversion. And I'm a better person for it. And
I
I'm,
agree.
I'm
I've
worth,
known you for a long time and you've always been wonderful, but it's it, it's a marked difference.
yeah, I like myself better now. I still sin every day, but unfortunately, I'll never stop sinning. But at least I pray for forgiveness.
Well, I knew you back then after your first wife died, and I really thought um, that you were on your way to death. To be honest with you, I was very concerned about your health and your. It's like you'd lost your fire, and I'm glad to say that since that time that you've made a remarkable recovery, and I thank God for that every day. That you've got your fire back and your desire back, and I'm thankful for Cheryl, and I'm thankful to God for that spark coming back in you.
We live in a world where we have evolutionarily conserved molecular pathways. What does it mean if the pathways in a walnut tree and a tadpole and a paramecium, and it's all the same? It's no different in a man or a woman or a human or a child. It's the same in all creatures. Same management of sugar, same management of DNA, RNA, same policy. Where did it all come from? It wasn't evolution, because evolution doesn't stop. There
Mm.
have been lawsuits about teaching creationism, and I've not been called as an expert in there, but uh, I'm convinced that evolution just doesn't make sense when we think that Charles Darwin could shoot little finches and reconstruct a theory in 19 days out of 36 spent on the Galapagos Island, collecting all these dead birds and taking them back to his current place in the country. His laboratory is moldy, by the way, because he had chronic fatigue syndrome, would leave his laboratory to go get healed. But it made no sense to think that could happen. Evolution, survival of the fittest, sure, that's what they're talking about. But let's explain where these pathways that are evolutionarily conserved come from. In the meantime, with the one-on-one relationship with a higher power, makes you keep your toes on straight, makes you think, makes you love, makes you care, make what's well the time we have on this earth worthwhile. And I'm enjoying the fruits of that. I'm the happiest man in Pocomoke City.
Awesome. That is so awesome. And I'm so happy for you that you found that. I mean, I've always been very impressed with you, Dr. Shoemaker, as a scientist and as a human and as your intellect, always searching for the next chunk of truth. Um, but I think this is something that, as it's been added to you, as you've received it, has really completed you as a human being. And yes, we still have a ways to go and all that, but I couldn't be happier for you and couldn't be happier for Cheryl. So thank you very much for the time and over these 6 segments. And I just like to leave it as, is there anything that you'd like to talk about, anything that you'd like to bring up before we close out this series?
Well, thank you for the opportunity to bring out some very personal feelings and very personal changes in life structure. You've been a role model to me. You're a minister, and you always knew something I didn't know. And I'm striving to do better. I'm striving to learn more. But I thank you for your courtesy over the years you've heard. I thank Cheryl, who's made my life a delightful place to be. Thank God every single day.
Well, I think that pretty well sums it up. So thank you again, Dr. Ritchie Shoemaker, the person who has discovered and parsed out almost everything that we know about chronic inflammatory response syndrome. Thank you for your dedication over the years and your self-sacrifice. And thank you for the time that you've poured into me and to many others over the years. And thank you for the time to allow this interview. We just appreciate it. I can't thank you enough, Dr. Ritchie Shoemaker. And for those of you who have been watching, to me, this has been a compelling series. I hope you've watched all 6 episodes of this interview. Dr. Shoemaker is an amazing man, and I'm so thankful that he's been with us. So God bless you. Have a wonderful day, and we'll talk to you soon. And I'll see you in Tempe.
You're right about that.
Awesome.
Thank you. Thank you so much.